Signal transduction can take place in liver cells and contains a hormone called epinephrine. Epinephrine is also known as the ligand. The ligand is attached to the metatropic receptor, which is in orange, and it needs attached to the receptor to get into the cell. The metatropic receptor binds to the ligand and goes through a process called protein conformation, which then changes the shape of the protein. Located underneath the metatropic receptor are the G proteins. The G protein consists of 3 subunits, the alpha, gamma, and beta. The alpha subunit has GDP attached to it. The alpha subunit then swaps GDP for GTP through a process of phosphorylation. The alpha subunit seeks out adenyl cyclase, which is in yellow, which is an inactive enzyme, but when the alpha subunit attaches to the adenyl cyclase, it becomes an active enzyme and is ready to convert ATP into CAMP. The process of phosphodiesterase is performed in order to get CAMP from ATP. In this case, 2 phosphate groups are taken away. The enzyme that speeds up the process of phosphorylation is phosphodiesterase enzyme. The sugar molecules create the cyclic portion that is attached to the CAMP. This is considered a secondary messenger. These CAMP are spread throughout the cell. CAMP will find the protein kinase, which is the red and the yellow, and it's made up of 2 subunits, a catalytic portion and a regulatory portion. The CAMP binds on the regulatory portion and as a result will activate the catalytic portion and the catalytic subunit will detach. When the catalytic portion detaches, an ATP molecule binds to a catalytic subunit. The catalytic subunit will bind to the phosphorylase, and the phosphorylase becomes active. Phosphorylase breaks down the glucose in the cell that can be released outside of the cell. The cellular response to this process causes an increase in heart rate, dilated blood vessels, and much more. Of the membrane receptors, G protein, adenyl cyclase, and signal transduction is you're going to have an epinephrine hormone, which is also known as the ligand. And what the ligand is trying to do is to get into the metatropic receptor, but in order for it to do that, it has to undergo protein conformation. And basically what protein conformation does is it changes the shape of the metatropic receptor, and then it then allows the ligand to enter in. And then underneath the metatropic receptor, you're going to have G proteins. The G proteins consist of an alpha, beta, and gamma subunit. And the alpha subunit contains GDP. But then the alpha subunit is going to hitch off of the metatropic receptor. And then while it's doing that, it's going to then replace GDP for GTP. And in this process, it's trying to get to the adenyl cyclase and produce ATP. And then once ATP is produced, it's going to turn into CAMP. And it's going to go into the protein kinases A, which contains the regulatory and the catalytic. And CAMP is then going to attach to the regulatory. And since it's attached to that, it's going to have the catalytic is going to come off because then it's going to have ATP attached to it. And then while ATP is attached to it, it's going to undergo a process called phosphorylase. And basically what phosphorylase is going to do is it's going to allow glucose to exit the cell membrane. And what the cellular response to this is going to be is it's going to give the body an increase in heart rate.